This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1261, recorded on October 10, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from College Station, Texas, Jolene Ramsey.
Hola. It's good to be here. It's a nice, warm Texas fall day, only 85 degrees Fahrenheit and 29 degrees Celsius.
Here it's 17 C and it's cloudy. We are expecting nor'easters this weekend. It's going to rain Saturday, Sunday, Monday, Tuesday. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's just a couple of degrees short, just a degree short of a palindrome here. It's 59 Fahrenheit, 15 C. And we are got beautiful weather today, beautiful fall weather. The leaves are just starting to change. A couple of weeks we'll get peak foliage. And yeah, same northeasters coming through here. We're far enough inland that it usually doesn't slam us too hard, but we'll see.
We're not too far from the water.
Yeah.
Here in New York, the water's
No,
right.
no, you're in a harbor city.
Harbor, isn't that how they say it in your state?
Yeah, Boston harbor. But Springfield is way out of it.
It's way out, yeah. You don't have to worry. I just learned that the latest Mac OS has widgets. You can put the weather on your desktop.
Oh.
You know, it's called a widget, which other OSs have
That's
had
a new thing?
Yeah, it's apparently a new thing. Well, I just discovered it, right? Maybe it was there.
Mac OS, even back when I was using it a decade ago, it had widgets.
So you could put the weather on the desktop?
You could put it, it was like a screen, like a transparency that could come down. I played with them a little bit.
There's a thing you click on the top right and it'll pull a little menu out where you have all kinds of things.
I see.
But I don't use it on the desktop, so I wasn't aware.
But I'm working on my laptop, and I always pick up my phone to look at the weather, but now I don't have to.
Yeah.
So but there are other widgets you can put there, too. You can put a calendar. You could put anything you want. I thought that was unusual, but it just shows that I never look at features. It's okay. Yeah, so it's fall. It is fall here. If you'll enjoy these programs where we talk about science. So the purpose of TWiV and all the other podcasts is to talk about science, try and explain it to people who may not be scientists. So we take the papers and discuss them. That's real science. And, we tell you what the authors think. We tell you what we think. But we don't lie about the results. We don't take a paper that says there's, no connection between Tylenol or paracetamol and autism and say there is a connection, right? In fact, once we did a paper saying COVID increased cancer, and an epidemiologist wrote in and said, not really.
Mm-hmm.
So we have self-correcting mechanisms.
Yep.
We have exonucleases who are outside.
We're not always right, but we are always curious.
We're curious, and we do our best to present science in a correct way. But anyway, if you like that sort of thing, and I know it's not for everyone, many of you like a 1-minute summary, which is great as long as it's correct. But we do excerpt summaries from our podcast, so you can find those on all the social media. But if you enjoy this, we'd love your support. That's a long way of saying we'd love your support. And you can go to microbe.tv/contribute. It's funny. When I was driving years ago, I listened to the radio and I couldn't take the ads. I just hated the ads. So then I started listening to NPR. And, every so often they have a fundraising. And I got tired of that. And now we're fundraising. So I started listening to podcasts because there are no ads. And now we're doing a little ad.
Yeah, but we never do a whole episode that's 2 hours of telethon,
Oh, is that what they do on
NPR?
Yeah.
At least I haven't actually listened to them on the broadcast channel in a long time.
Well, now they're under pressure to replace.
Well, yeah, now I feel bad but we give money to them.
You may not remember, Jolene, but Alan was around when we did actual ads.
Yeah.
Here on
I
Twitter.
remember. I do.
Yeah.
The Drobo.
Oh, yes, the Drobo. We gave Mark Fuccio, who used to work for Drobo and still listens, he gave us a few units and we gave them away. We had some contest.
I had mine backing up my office for years. I've moved to a different solution now, but
yeah, in fact, Dixon had one, and so I was at a meeting years ago, and someone came up to me and goes, "I won a Drobo."
Yeah,
that's cool. The Drobos were very good.
Yeah, they went under, I think.
And also we did ads for Blue Apron.
Yep.
Yes,
I tried them.
I did
too. And I was amazed at how many scientists tried them and said, "This is perfect for me because I don't have a lot of time." And it is very easy to cook with that, but of course, many people like to do their own thing, right?
The only reason I stopped was because I like leftovers, and it was so proportioned that there weren't leftovers.
True.
I didn't like the waste disposal problem.
There was a lot of stuff, right?
They sent this huge pack of packaging, and they would, they had this little kind of, I don't know, they would say in it, be sure to dispose of things properly. Like they're just washing their hands of the fact that they sent you this big box of plastic.
Yeah. And I know that, remember, they would give you soy sauce. They give you a little plastic
tiny
bottle.
Yeah.
And then you'd throw it, you generate a lot of waste, that's for sure. But, we were ahead of our time when Blue Apron just came out. They advertised on us, and I'm sure they did. We did Curiosity Stream, which was
I tried that too.
A streaming service for like.
But now we don't do ads. We just do recollections of ads.
Isn't that funny? We're recalling. Well, that's my fault. But yeah, it's. In fact, the ad company we were using, you need to use an ad agency mostly, because otherwise it's too much work to.
Yeah.
And the ad agencies typically take 30%, right? And that's a lot of money. But also the ad agency emailed me one day and said, we don't make enough money off of you. We're getting rid of you. The lady who ran it was very blunt.
Okay.
I said, okay, then we're not going to do ads.
Yep.
And I decided not to, and that's, around the beginning of the pandemic is when our YouTube started to grow, and I don't want to do ads there either. So here we are. We're depending on your support, and so far it's worked.
Yeah.
And it should work because you're very generous, and I appreciate that. We appreciate it, but we want more people to listen because the people are getting misled by what do you call those people?
Liars.
Influencers, yes, liars.
Oh.
Liars. Actually, people get influenced by the wrong people, and it's unfortunate. And then they say that we are liars.
Right, gaslighting. Right? Yeah.
What is it? Someone told me, someone at a hearing, someone said to RFK Jr., you said this. And he said, no, you're lying. Something novel, right? That's what they accuse. Anyway, they have few news items, and I think, Alan, did you pick that first?
Yeah, we actually have some positive news today, so this is
all positive. Yeah,
a very nice thing. So the item I picked is the winners of this year's MacArthur Award, and I was scrolling through those, and I saw this name. I said, that sounds really familiar, and they described his work. Jason McLellan, go listen to TWiV 1103. Just won a MacArthur Award, sometimes called a Genius Award. So he got a lump of money from the MacArthur Foundation for his work. And congratulations, Jason.
Someone asked me on the stream the other night, so how many MacArthur Award winners have been on TWiV? That's a wonderful question.
Got one now.
So Jason was on.
Yeah.
Nels Elde has been on TWiV.
Yeah.
Because he has his own podcast. And also Trevor Bedford. Who was on ASV TWiV. And there are some others as well. I can't remember them.
See, the TWiV bump can take you really high.
I'm not sure if the correlation is quite strong enough.
Oh, I'm sticking with it.
All the people who work on viruses that have won MacArthur Awards, and then that might be stronger.
Yeah.
Yeah. So anyway, it's nice that we pick interesting people, right, for TWiV episodes. That's what I like about that. So Jason was on 1103, 714, and 500, in which we all went.
That's right.
To Austin, and you went to that, Jolene. Right?
I did.
You drove like 3 hours to get there.
2.
More.
And again, we did possibly perhaps get stopped along the way.
Yeah, that's right. You say that all the time. 714 was online. That was during COVID. And 1103, Rich and I had gone to Austin for that. Well, he lives there.
Yeah.
I went for South by Southwest, and then we did that with Jason while I was there. And then we also did TWiV 100 at the Science Mill, which is a science museum outside of Austin. It's not really outside of. It's in Johnson City, which is quite a drive from maybe an hour. Does that sound right?
Something like that.
But in Texas, that's not much of a drive, right?
Actually, that's half of a drive.
It's half of a drive. And I learned that Johnson City was named after LBJ's, I don't know, grandfather or something who founded the city. Very interesting stuff. I have some news here. Well, this is very common news, but I wanted to talk about it because it's really cool. The Nobel Prizes have been announced this week. And oh my gosh, today the Peace Prize was announced. Thank goodness.
Yes.
A lady from Venezuela. Oh, how nice. I'm so happy. You know, I knew the Swedes would do it right because they're very serious. Thank you very much. Anyway, the Physiology or Medicine Prize went to Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi for discovering regulatory T cells. And the story is so beautiful. I encourage you to go to the Nobel website where they have wonderful descriptions of the prize winners. They have both a lay version and an advanced science version. And this story, I'll just briefly tell you this story. So Sakaguchi basically took the thymus out of rodents and found that they got autoimmune diseases, okay? And he identified a population of CD4 T cells that also produce CD, I forgot what CD it is. It's maybe 45. And those are the population. And he called them regulatory T cells or Tregs, all right? And independently, Brunkow and Ramsdell, they're working at a company on the Pacific Northwest, and they were working on scurfy mice, which were mice that had been irradiated. They're looking for mutations, and this scurfy mice developed autoimmune diseases. So the company allowed them to identify the mutation in a single X-linked gene, which they named FOXP3. And then Sakaguchi goes, oh, he says, this is missing in T cells from thymectomized mice. And then Brunkow and Ramsdell then show that in the scurfy mice, there's a mutation in the gene and it prevents the production of the protein. And then finally, somebody else who didn't get the prize knocked it out and showed that you got autoimmune diseases. And then one of these also showed if you put the FOXP3 gene into normal CD4 T cells, they become Tregs. So it's a beautiful synergy between these individuals, and it's important because Tregs are medically important. You know, if you don't have enough, you get autoimmune diseases
like the mice.
You have too many. Like in a tumor, you need less Tregs so that the T cells can go crazy in the tumor. So there are therapeutic developments from this as well. So I think it's a wonderful Nobel Prize.
Yeah,
Yes,
good pick.
Congratulations to them.
Yes.
So Mary got her PhD in Princeton. Fred got his PhD from UCLA. And Shimon got it from Kyoto University, which is a lovely town. Just amazing. Also, I wanted to show you a BBC article about the recent success with treating patients with Huntington's disease. And it's written very nicely, right? That's why this is by James Gallagher, and it's from back the 24th of September. But, you know, in Huntington's, the patients degenerate over time and they can't be saved. But now what they did was that we know that in patients, the Huntington gene is mutated and the protein is dysfunctional. And so what they did, this is not a trivial treatment. They have essentially viruses carrying microRNAs that target the Huntington mRNA. So you make less toxic protein, right? But you have to infuse it into the brain. Apparently, it's like a 12-hour surgery
Right.
Yeah.
to make a hole in the right place and all that. But they had in the patients 75%. The disease was slowed by 75% in the patient. So basically, they say the decline you would normally expect in a year would take 4 years after the treatment. So
Yeah.
it's good.
And that's, it's not a super fast disease, but the onset, I think, is usually in like late 20s, early 30s.
Yeah,
That's right.
mid-30s.
And then within a couple of decades, people are dead and, and they're kind of falling apart in the meantime. But if you have a 75% decrease in the speed of that, it's not a cure, but at a certain point, if it's going to take 80 years for this to set in, then it kind of is a cure because by that point you're,
right.
probably not around anymore. So it has not been, we should note, this has not been peer reviewed and published yet.
No, it's just an announcement by the company.
The clinical trial, right?
Clinical trial,
Basically,
clinical trial results.
Trial, yeah.
Also, it's projected to cost $1 million a person. So it's...
Because C note about 12-hour neurosurgery.
Yeah.
That's not a bad price, actually. There are other gene therapies that are more,
right?
That don't even involve brain surgery. They just involve the infusion. So, it's
a major advancement, though.
Yes,
they really had nothing before.
Yeah. And the fact is, a proof of principle also that you can slow disease progression by taking out this protein. So anyway, that's very cool. All right, on to the literature. Now, here's how today's episode developed. So midweek or early week, Jolene emails us and she says, I'm going to pick this paper. And I had seen that paper and I thought it was really cool. And I was going to email you like on Sunday and say, do you think this is cool?
I picked it the day it came out.
So she picked it. And then I was looking. I saw the title and I didn't read it, so I didn't know exactly what was in it. But you'll see what's in it. But then I saw an amazing paper where they did the cryo-EM structure of an archaeal phage, and the figures just blew me away. That sold me immediately. They're so beautiful and clear, and then the biology is interesting. We don't really talk much about archaea, and so I thought. We did years ago when Ken Steadman was on, who studies an archaeal virus that the archaea grows at very high temperatures, so he can't put water in his water baths. He puts ethylene glycol, right, antifreeze, so it won't evaporate to grow his cultures. But Ken is very interesting. He walks around meetings with a model of his archaeal virus. It's this ovoid, elongated thing, and he's always got it with him. But apparently, according to this paper, it's not a complete model. So anyway, that's how we arrived here, and you're going to learn a lot about bacteriophages and archaeal viruses.
Yes.
So the paper, the snippet is in Science Advances. Cryo-EM resolves the structure of the archaeal double-stranded DNA virus HFTV1 from head to tail. Alan, you want to do the authors?
Yeah, first author is Daniel Zhang, senior author is Bertram Daum, and the group is from University of Exeter, University of Groningen, and University of Helsinki.
I think this was open access.
Yeah,
right?
both of the papers today are open access. Both are actually in Science Advances.
So, as everyone knows, viruses infect everything, including archaea. Now, archaea are one of the three domains of life. We have archaea, prokaryotes, and eukaryotes, right? And archaea are very interesting because many of them look like bacteria, but they don't have 16S ribosomal RNA, right? That's what
when I was in college, this was called archaebacteria.
Archaebacteria.
And then we found out they're not bacteria. They're not
bacteria.
Remotely.
When you say they look like bacteria, you mean like the size of the cells.
Yeah.
Right.
Yeah.
Other than that.
They don't look like a eukaryote, right?
Right.
But yeah, then biochemically and genetically, they're I guess they're closer to eukaryotes than they are to prokaryotes, but they also have these unusual lipids, ether-linked lipids in their membranes.
One of the reasons it took us a long time to figure these guys out is that they tend to grow in extreme environments. And they're often not things that you can just, pop in a broth and grow in the incubator. As Vincent said, you're either boiling these things in polyethylene glycol, or in this case, it's a high salt archaea that they're dealing with.
But there are certainly some that are not extremophiles.
There are some that are not extremophiles.
They're in our guts too. I think that's important for people to know. Point. There aren't any, although there was a report that maybe there might be one associated with human disease, but otherwise they don't cause any diseases that we know of.
How interesting. They're the nice microbes.
Yeah.
Anyway, they're everywhere. They're very important. I guess they're in cows, and that's how they make methane, right?
Yeah.
Anyway, they have their own viruses. We don't call them bacteriophages because they're not bacteria. We call them viruses. And they have a variety of shapes. They can be spiral. They can be bottle-shaped. I think Ken's virus is bottle-shaped. They can be. Oh, no, his is spindle-shaped.
It is, yeah.
Spindle-shaped.
I was going to say, if it's bottle-shaped and he's always carrying it around, you might have some concerns. Maybe that's not a virus model.
The really common ones are viruses with a icosahedral head with double-stranded DNA and a helical tail. These are tailed viruses, or TVs. So now we have the TV. There you go. It's on.
Yes, it's on my. There's a tailed virus on my pillow. Thank you.
Too. These are made by Jolene's mother.
Jolene's mom.
That's right.
This is called needlepoint. No, it's not needlepoint.
It's quilting.
Quilting. Yes.
Yeah. Yep. It's a paper piece, actually.
And so, we find a lot of those in bacteria as well, but also in archaea. And so the archaea ones are called ATVs. So now we have TV, which may end up in the title of this episode, because you will see that Jolene's paper also deals with a TV. And
these are archaeal tailed viruses, not all-terrain vehicles.
Tailed viruses. It's interesting that bacteriophages and the archaeal phages, the tailed phages, have this common architecture. So there's probably a common ancestor at some point a long time ago. But anyway, this paper is very cool because it's got beautiful, they solve the structure of a particular archaeal virus, and it's just beautiful. But it also sheds a lot of light on mechanisms of infection. So they have an icosahedral head, which or capsid, which contains the double-stranded DNA. Then they have a tail, an extended tail. Now, this tail could be different lengths. Those of you not in science who just think about bacteriophages or look at our quilting, you may think they all have long tails, but they don't. They can have different tail types, right? They have long and medium and short tails. I think your phage has a short tail, right, Jolene? The one in your paper.
The one, yes, that's right. Yeah,
yes.
They have the short tail, and the longer ones can be flexible or rigid as well. Right. Contractile, meaning they can actually rearrange and inject like a needle, or the flexible ones that don't do that.
So the tails are involved in, there's typically a base plate on the tail, and it's involved in recognizing the host, and the DNA goes through the tail and into the host. We'll talk about that as well. And so these viruses inject their DNA. In this case, the archaea, it's interesting. These capsids are made of proteins, multiple proteins. There's a main capsid protein or major capsid protein, MCP, that forms the capsid. And then there are scaffolding proteins that help build the capsid on assembly, and those are gotten rid of, just very much like herpes viruses, where there's an internal scaffold built and then it's degraded. And then there's a portal protein put on the head. Same thing with herpes virus. There's a single portal protein. The genome's loaded in it, and then on infection, the genome comes out of the portal. And for these tailed phages, the portal is where the tail is attached. The DNA is put into the capsid in an energy-dependent mechanism. There is a what's called the terminase complex there on the portal, and that is an energy-dependent mechanism. For eukaryotic viruses, they've been very well studied also, and they need energy, and they pump the DNA in under very, very high pressure. For the herpes viruses, it's like 600 pounds per square inch. And I tell my class, just think about your car tire or your bike tire. It's less than 100 PSI.
Yeah.
600 is a lot. And why is that? So that when the opening comes open, the DNA shoots out under the right conditions into the cell. So we're going to look at that in this. We're going to look also at how the DNA is packaged in the capsid. And in bacteriophages, it's been shown that the DNA is typically in coaxial spools. You can see very nice EMs of that within the capsid. And these are typically aligned with the portal. And we're going to see what happens here. So nobody has studied a lot of archaeal TVs very much. We know something about the receptor. So 2 receptors have identified. There's a layer on the outside of the archaea called the S layer, made up of proteins, carbohydrates. It's a major envelope component of archaea. And so there are 2 archaeal viruses that have been shown to attach via this. And one of them, Halophorax TV1, HFTV1, which is the subject of this, first attaches to the host via the head and then the tail, which is not something I knew at all. I don't know why I didn't know that. It's a testimony to my ignorance.
Well, there are so many viruses to keep track of. I don't think you can be expected to know everything about everything.
I love them, and
Yes.
I should keep track of them.
And now you know.
And now I know.
This one sticks its head and then its tail.
So the head, and you'll see how it attaches, and then the tail hits on, and then once the tail attaches, the DNA is injected into the host cell. All right, so they have purified. They're working on HFTV1. So this is Halophorax TV1, and the name should tell you that these are growing in hypersaline environments, right? Halophorax, and you'll see the reason I mentioned that. You'll see that in a moment. So they grew up these viruses in their host, and they purify them, and they noticed there are both full and empty particles in the preparation. So when they did cryo-EM to solve the structure of these viruses, they noticed that there are 2 classes. There are classes with and without DNA. So perfect opportunity to solve the structure of the pre and post ejection, which is what they did, and get a lot of information of what's going on here. I just want to mention one thing about, so they collect cryo-EM data, right? So the specimens are frozen in aqueous solutions, and they take many, many pictures of them in different orientations. And then they use a model protein, a model program on the computer, which appropriately is called Model Angelo.
Yes.
I didn't notice that.
Michelangelo, Model Angelo, right? It's beautiful. I love when people have sense of humor. So they show that, so it's 1,350 angstroms long. There's an icosahedral head. It's 600 angstroms in diameter. There are 60 turrets on the head, which are about 90 angstroms long. So 60 should ring a bell, because in particles with icosahedral symmetry, there are 60 5-fold axes of symmetry. And so there's a turret at each fold axis of symmetry. The tail is 560 angstroms long, and it ends in a base plate.
This would be a beautiful model to 3D print.
Oh, yes.
It is
yes.
gorgeous.
It's a beautiful, the images they have in the paper are gorgeous. And that would be the whole thing, right? With the head, the tail, and
all
the base plate.
I'm thinking somebody on this paper has probably done that.
They have. I had poliovirus with its receptor 3D printed. It's right up there. It's gorgeous. And the guy I was working with said, we should crack it open. So he put magnets in it so you could actually take it apart. And that was $450.
Really?
So I'm guessing this is going to be $900.
Wait, but how big is it?
I'll show you. It's quite big.
Oh, yeah.
Yeah.
Yeah, it's quite big.
It's like, what, 8 or 10 inches?
No, it's not that big. Look at my head.
Eh.
My head is not that
It's
big.
not smarter than you. Its brain is smaller.
Is that from a filament or a resin printer? It looks really detailed.
I have no idea.
Okay.
but he said we have to put a plaque on the front that says poliovirus plus PVR, and then it opens up.
Oh,
Right.
wow.
So, and the interior is correct. It's the structure. We don't have any RNA in here, but I do have a beaded genome behind me. I could stuff it in there. But yeah, the phage would be beautiful.
Yeah, but it would be under too much pressure. It would explode.
All right, so that's the phage, the virus with DNA in it. We'll talk about the DNA in a minute. Then the empty virus, compared to the one with DNA, is missing the tape measure protein, which Jolene talked about a while ago.
Has two functions. Tape measure protein is for determining the length of the tail, and also functions in entry, helps with getting across the membrane.
Base plate hub and the tail fibers. So there's the base plate and little tail fibers coming out from the base plate. So those are gone. And so the idea is that when the DNA comes out, all of those things, the tape measure, the base plate hub, and the tail fibers come out. The tail is the same length, so it's not contracting. As Jolene said, some of the phages contract, but not this one.
Wait, did they say that the tail fibers come off when the DNA comes out?
Yeah, they're missing in the empty capsid reconstructions. Yeah.
Okay.
They say they're jettisoned. I like that word. And the heads are the same, so there are no structural changes in the head when the DNA goes out. So this is a T equals 7 icosahedron. T is the triangulation number, which means there are 7 protein subunits in the structural unit. But what's cool about this, it forms a right-handed or dextro icosahedron, which is highly unusual. Not many viruses have T equals 7 dextro capsids. File that away in your knowledge. It will be useful one day. About right and left-handed chemicals, right? So it's the same idea for the capsid. So this is a very cool capsid. It's made up of 60 copies of the asymmetric unit, which is made up of other proteins, including the major capsid protein. And because it's more than T equals 1, it has pentamers and hexamers. So T equals 1 viruses have only pentamers. I can show you that. Look. I'll show you. I can't really. I only have a T equals, I think this is a T equals 3. So you can see there are blue and there are red subunits. So the red are pentamers. There are 5 of the capsid subunits associated together. And then actually this cyan is the hexamers. So when you make anything bigger than T equals 1, you always have to have these pentamers and hexamers. And in the virus in this paper, it's fun. It's hard to get pentamers and hexamers to associate with each other. It makes it a little less stable than the T equals 1 virus. But in this virus, there are trimers that help the two interact better. And structurally, this is very much like the major capsid protein is very much like that of tailed bacteriophages. And in fact, it has a classical fold called the HK97 fold, which was first discovered in the HK97 bacteriophage, and since it's been found in many, many other viruses, including herpes viruses. So it's an ancient way to fold a capsid protein, and I shouldn't be holding that up because that's not even related to what I'm talking about. The HFTV1 major capsid protein also has an HK97 fold. So
the HK97 fold, I hear about this all the time, and it's never really registered in my brain. I'm like, what is the HK97 fold? I know it's everywhere. Everybody's like, oh, it's HK97. Like, I'm like, oh, yes, of course. I totally know what that means.
Well, it's a way to, it's a way that
it's an arrangement of the major capsid protein that's common between many of them. But I actually looked it up so I could see what the features of the HK97 fold are. And it's not just been seen in low T number viruses, but also I saw up to a T equals 52 jumbo phage, where there's more than 3,000 monomers that are used for making the final capsid. And
Wow.
it's a nice fold with very distinctive features that are found common among the HK97 fold proteins. So there's a couple of nice reviews for anybody interested in looking at it.
Now, the parts, remember, the major capsid protein makes this closed shell. So there's an interior surface. And when you look at that, it is negatively charged. And they speculate that, well, DNA is negatively charged. And they speculate that that prevents the DNA from sticking to the capsid, so it gets out easier. And that makes perfect sense. Okay, so
it's a non-stick coating on the inside.
Non-stick coating. What are they called?
Teflon?
Everywhere chemicals.
Yeah. Oh, right. Yes. Forever chemicals.
Forever chemicals. Yeah.
Only in this case, it's just done with charge, so it's biodegradable.
Now, one of the things that's interesting about this virus structure is that there are a lot of magnesium ions everywhere. So, for example, there's this major capsid protein, and then there's the trimer that stabilizes the major capsid protein. There are lots of magnesiums at the interface, which they say probably stabilize the interface. They're actually going to do an experiment later to prove that. And there are many, many magnesiums elsewhere, which probably they say is because this thing grows in high salt environments, right? So it was selected to be able to do that. The other aspect that's very cool is that you can see the DNA. Oh my gosh, you can see the double helix of the DNA inside the capsid. The resolution is so good.
Yes.
If you look in figure 3, the DNA is in purple. Mm-hmm. I've never seen anything like this, although it probably exists. It's just beautiful. It's arranged in 10 concentric shells. And they can see the ends of the DNA, right? There are 2 ends on this long DNA. One of them is in the portal, right? It's the entry-exit way in the capsid. And then the other, they say, is probably in the outermost shell. And lots of other information about that.
So it's like this tightly wrapped ball of twine.
Yes, it's tightly wrapped. They say the genome sequence has some DNA binding proteins predicted in it, but they don't see any in the capsid. They don't just see DNA. So what gets the DNA in there and so forth isn't known. But what they do notice is that both DNA and double-stranded DNA and double-stranded RNA viruses have this spooling of the nucleic acid in the capsid, even though they package them very differently. So they say this spooling must be good for something, probably for ejection, right? Maybe for packaging too. It's very efficient to packaging it that way. So they have a mechanism for spooling that they propose. They start from the portal. The DNA winds upward to form the outermost shell. And then at the pole opposite the portal, it goes into the second layer and spools back down to the portal. And then this is repeated 9 times, and that gets all the DNA into the particle.
Yeah, so it's like a ball of string, but it's been wound up from the outside in.
Right, exactly. Try doing that at home.
Not easy.
Now, the turrets, this is what got me to do this paper. I said, the turrets on an icosahedral capsid. The turret is made up of several components. So I'm going to go to the picture here. So we have 2 proteins. There's a base, which is a hexamer of a protein called GP30. It's called the turret base protein. Good name. I like that. And then there's a trimeric turret head protein. So basically, this whole thing looks like a mushroom, and the turret base protein fits into the hexamers of the major capsid protein. There's a little stalk there. And then the turret head protein fits into the turret base protein hexamer. Okay. And if you look at the picture, you can see that beautifully. It's lovely. There's the head, there's the base, and there's a little neck between the two, and then that fits into the hexamer. Now, this protein, the turret head protein, right? This is presumably how it's attaching to cells. They say it has protein similarity with polysaccharide deacetylases from multiple species. And so these would be proteins, deacetylase enzymes that bind polysaccharides, so that maybe that's how this thing binds to the cell initially, to that S layer via that activity. Something you could test, right?
Right.
All right, then we go to the portal. The portal is made up of a protein called GP13. It's a dodecamer. That's 12, right?
Yep, that's 12.
Dodecamer of GP13. And it goes through the whole capsid at one 5-fold axis of symmetry. And there is a symmetry mismatch there, right? Because the portal is a dodecamer, and then the capsid is a pentamer.
Mm-hmm.
So how do you do that? And there's an intermediary protein that can bind to both. It's the portal interface protein. That's the other thing I like. Every protein has a great name. Right?
They could have called it the gasket, but...
The gasket. So GP13 was back when the genome was annotated and they just numbered because they didn't know what they do. But now they have functions, so they're giving them lots of names. Yeah, this is the PIP protein. So that's very cool. Again, the portal is probably where the DNA gets in. In a mature virus particle, they can see the end of the double-stranded DNA in the portal, protruding through the central channel of the portal protein. And this channel is lined with phosphohistidine residues. So they would have a negative charge on the portal tunnel, and again, make so the DNA doesn't stick to it as it's going out. Teflon again, although it's not Teflon. That's a fluorine-derived thing, right, Alan? But this is very, very clever. Evolution is just amazing. And again, that if they compare the full DNA full and DNA empty structures, the portal protein doesn't have any difference. So there's no conformational changes in the protein, at least none that are detected by cryo-EM.
Did they mention if the phosphohistidines are still phosphorylated in the empty particle?
Let's see. This is the mature particle. They don't mention that, no. They don't mention that.
I ask because I did a little bit of reading about those, and it's a pretty unstable modification, unlike when other residues are phosphorylated. So I don't know if the conditions that they did their structure in would maintain it, but I guess because it's cryo, it could.
Yeah, right. And then we have the tail. Another figure 6. So beautiful.
Yes.
Mm-hmm.
Because they've also colored them beautifully, which really helps. If it were all one color, then it wouldn't be as stunning.
Yeah, but the level of detail on these structures is just spectacular.
You can see the tape measure protein goes from the portal all the way down to the base plate, and it's alpha helical protein, as you might expect for an extended protein. So this tail starts with a protein GP26, which is called the head-tail connector, or HTC, right? So that holds the portal to the tail, and the DNA can pass through it. Then we have the tail completion protein, GP29, and 11 copies of the tail tube protein, TTP, and that makes up most of the tail. It's a hollow tunnel, 36 angstroms in diameter, and it houses the tape measure protein inside. So the tape measure protein is in the lumen of the tail. And the tape measure protein goes from the first tail tube protein near the portal to a cavity in the base plate hub, the base plate hub, BPH. And they note that the coiled coil region of this tape measure aligns perfectly with 11 tail tube rings, so that they say this confirms that the tape measure protein dictates the tail tube length, as Jolene said before. Then there's a protein between the tape measure protein and the DNA, which they call the TMP DNA spacer. And both the tape measure protein and the TDS, the DNA spacer, are gone, so they're released during DNA ejection. Then there's the base plate hub attached to the bottom of the tail, and this anchors 3 tail fibers. So you can see that very nicely in the picture. They're not very long. They're extended with a little blob on the end. Maybe not like the tail fibers in our pillows here, but much shorter, but still beautiful. They're highly flexible, which they say is good for docking onto the cell. The base plate hub has structural similarity with xylinases and glycosaminidases, right? These are enzymes that would cut various sugars. So maybe, again, they play a role in binding or maybe even degradation of the S layer. They note that the tail is long enough to span the S layer to the membrane. So it could be that these degrade the S layer and then allow the base plate to sit on the membrane, but they don't know yet. So the magnesium's everywhere. I haven't mentioned most of them, but they do an experiment by removing magnesium from virus particles using a chelating agent, and now they're no longer infectious, and the virus particles fall apart. So the magnesiums are important. I'm going to start drinking magnesium because I heard it helps you sleep.
You heard?
Well, many people have told me.
If you drink enough of it, then you become flammable.
I did look it up. I looked at some articles, and this is the collision between science and society, right? If you read the science, they say the data is not great that it helps you sleep. But if you talk to people, they say, "Oh my gosh, I never slept as well in my life." Maybe it's a placebo effect.
Yeah.
I don't know. But we're going to see, because I don't sleep well, and I want to try and work on that.
Okay.
But that's just
as long as it doesn't harm you.
Yes.
Yeah, it shouldn't harm me.
If you wake up and there are sparks coming from your fingers, then
but
go easier on it.
If it doesn't work, I'm not going to use it, of course.
No, right. And it may make you susceptible to archaeophage.
I already have them.
Well, right, but this one in particular.
Okay, and then they do some comparison with other similar bacterial viruses with the head and the longish tail, and you can see a lot of similarity. And they say that HFTF1 is evolutionarily related to the tailed phages, the core structural proteins, particularly those on the portal, the capsid, the tail, the base plate, tail fibers. They could be products of genes horizontally exchanged between bacterial and archaeal viruses. That would be very cool, right? So those are the data. And then they have in the discussion a nice model of the attachment. They say that probably the virus attaches reversibly to host glycans via these turrets, and then maybe the enzymatic activity of the turret can modify the host glycan matrix. There's a very nice figure 8 where they're showing this happening. And so you have your S layer, which is glycosylated. The head is binding and possibly removing, and then it flips around, and the tail plate is binding either to the S layer or perhaps below it to the membrane of the virus.
Of the
host. And so now the tail is perpendicular to the membrane, and it can penetrate the membrane, and they say maybe the tail penetrates the membrane and the DNA then shoots out into the host cell.
So in bacteriophages, the ones that don't have a contractile tail that would be analogous to this, there is potentially some piercing of the membrane by a protein complex, but then the tape measure protein is what comes out and the DNA follows. The whole tail itself wouldn't be expected to pierce the membrane. I think part of the disadvantage of doing it that way would be then you have a hole that you have put into the cell. And in this case, this is the plasma membrane. So you don't want to have an unpluggable hole in your membrane if you want to make more viruses.
Right.
You know, interestingly, I didn't mention this, but they in one of their structures of the tail, so where is that? In figure 6. So the tail of the empty virus has a plug at the end of the portal, almost as if the syringe stays in the membrane and it's plugged so things don't leak out.
Right.
I don't know if that's true, but there is a plug. They have a question mark plug.
They just say a density is there, so they don't know what it is.
Density, yes. May not be a protein, right? Who knows? Anyway, so I like this because the figures are beautiful. It's a great structure. It's biologically interesting. And they say this structure presents a major step forward in our understanding of archaeal viruses, provides structural evidence of the strong evolutionary relationship between tailed viruses of bacteria and archaea, and opens new research directions to answer fundamental questions in virology, such as host cell infection and virus assembly. And I agree with all of that. I think that's great. And that's why this is a cool paper.
Yeah.
So I have one of those questions about the meanings of words. So in most of the paper, they talked about ejection of the DNA. And there's a couple spots where injection is used. And so my usage of the word prefers ejection if the DNA is coming out due to pressure, and injection if there's an active mechanism that's pushing it. I'm not entirely sure the best way to distinguish between them, but for the most part, ejection seems to make the most sense.
I would say, just from a word definition perspective, I think ejection usually means going out and injection is going in.
Well, then who's it relative to?
Exactly.
Yeah, right.
So you eject it from the virus and you inject it into the cell.
Oh, wow.
So when you're discussing it from the virus's perspective, when you're talking about the viral structure, you say this is for ejecting the DNA. But then when you're talking about the entry process, you say this is how the virus injects the DNA.
But I think that the differentiation comes when there are tails that actually are contractile and puncture through the host cell. And so I guess I was considering that the injection part, which this HFTV does not do.
Right.
So Jolene, when the tail is collapsing, does that provide the force to get the DNA in the cell?
No, it just punctures the membranes and gets through
So in that case, the DNA is still under pressure in the head, right?
Yeah, and it kind of depends on which phage you're talking about. They're not all as high pressured. It depends on the size of the genome relative to the interior volume. Some phage actually have to pull their DNA in using a polymerase.
Mm-hmm. So this is interesting because the herpes viruses have a portal. They have also an HK97 fold in their major capsid protein. So you can imagine there was a common ancestor. But for herpes virus, the virus packages the DNA through the portal with motors on the portal that put it in under high pressure. And then upon infection, the capsid docks onto the nuclear pore via that portal, and that opens the portal and the DNA shoots out into the nucleus because it's packaged under such high pressure. So
and it's ejected from the virus and injected into the nucleus, I would say. And
so similar mechanisms with these archaeal viruses and bacteriophages. So they're related.
But if your fighter jet is on fire, do not hit the inject button.
What would that do?
I don't know. I don't think there is one. The eject button will get you out of the plane, though.
Inject. Maybe it's like a fuel injector.
Right.
Like okay, so we promised you two. Well, they're not the same. This is a bacteriophage.
This is a bacteriophage, yes.
But it looks similar.
It's
right,
another tailed virus.
There are some themes in the structure. It's true.
So, Jolene, it's a T = 7 also.
Yes, it is.
I didn't know that when I picked this paper. This is just like divine intervention.
There are many more commonalities in biology and virology than we recognize on the surface sometimes.
Yeah.
So this paper is called "The Discovery of a Legionella Phage Explains a Key Determinant of Human Disease," also published in Science Advances in September of 2025. This comes from the group of Alexander Ensminger, first author Beth Nicholson from the University of Toronto in Canada. And this caught my eye because I've never really read anything about Legionella phages, and I guess that they kind of explain the reasons why. And it also has a beautiful structure in it, and it also has some themes in terms of like what is involved in getting phage DNA into the cell. So here, what the main kind of takeaway message is that they found a temperate phage of Legionella that makes virions. It doesn't make plaques. And they also found that a resistance phenotype that is selected by exposure to the phage is highly associated with the kinds of Legionella that cause human disease. So as a little bit more in-depth background, the Legionella pneumophila is a gram-negative bacterium that does cause human disease, although it's found in the environment and doesn't always have to cause disease. But when you are exposed through breathing in aerosols of water droplets with Legionella, where it tends to grow inside of amoeba, then you can develop an infection. And that infection in humans can range from a mild infection where the disease symptoms are fever, also called Pontiac disease, or it can progress to severe pneumonia, which is what we call Legionnaire's disease, after the epidemiological investigation of the summer after the summer of 1976, when there was a meeting of the American Legion where many individuals who stayed at the same place developed this unidentified pneumonia. And so they finally figured out that Legionella that was contaminating, I believe, the air conditioners had caused all of these individuals.
In Philadelphia.
Yes, in Philadelphia. And the original, so this was discovered twice by epidemiologists from the CDC. First was Pontiac fever, and they weren't able to isolate the causative agent. They had an outbreak of some kind of pneumonia somewhere. They weren't able to figure out what caused it, but the location of that was Pontiac, Michigan. Right. Which is why it's called Pontiac fever. It was not an ad campaign for a car. And then sometime later, 1975, there was the outbreak in Philadelphia of
76.
76. There was this outbreak in Philadelphia.
It's bicentennial.
Yes. And they managed to isolate the bacteria from that.
You put a link to a nice summary sheet, Jolene.
Yes.
Yeah, there's
it's really nice.
A microbe of the month, which gives all these kind of key facts about Legionella pneumophila, which is one of the major causative agents. There are some other Legionella species that can be associated with human disease, but the majority are caused by this serogroup 1. And that accounts for over 50% of reported cases of Legionnaire's disease in certain parts of the world. And fortunately, Legionella is treatable by antibiotics, and it doesn't spread person to person. It's usually by a contaminated water source, and so cleaning up that contaminated water source results in no more infections being propagated.
We had a sizable outbreak in Harlem, New York, this past fall and summer, which was traced to air conditioners.
Yep.
Where, there's always a little bit of water in the bottom of the air conditioner, and that's what does it.
Yeah. So do the right thing, clean your air conditioner.
You just clean it, and then it goes away.
And so humans are a dead-end host for this bacterium. It normally infects amoebas, and it can infect certain human cells, but it doesn't go anywhere from us.
Jolene, there's a sentence in the introduction that I just love. It says everything I've been working my whole life to explain.
Yeah.
It says, humans are an evolutionary dead end for L. pneumophila, and all fitness advantages in the human host are coincidental to selective pressures in the environment. So it's not intentionally getting more virulent for people. That's what people say. Well, the virus got more virulent. No, it's probably some coincidental change. I love the use of that word. It's perfect. One sentence, it's so clear. Poliovirus fits this perfectly. This is a virus that reproduces in the gut. That's it. That's its niche. And any selective pressures that affect fitness in the gut can coincidentally affect them elsewhere. For example, if the virus reproduces better in the gut, it becomes more fit. It's then more likely to accidentally invade the central nervous system. So it's not selected for. In fact, most neurotropic viruses, the CNS is a dead end, just like L. pneumophilus is a dead end in people. So there cannot be no direct selection, any direct selection there, which is something I've been trying to explain for years. And this one sentence says it beautifully.
So if we rephrase that, it would be the human central nervous system is an evolutionary dead end for X virus.
Right.
And all fitness advantages in the human host are coincidental to selective pressures in their environment / normal replication, which could be the rest of the human body, where it doesn't usually lead to that type of disease.
Exactly. Yeah. So the difference between Legionella, it's the real environment outside of the host, but for other viruses, it can be a different site within you. But yeah, that's it. I think it's all about fitness. The selection is all about fitness.
Yeah.
So even if you select for better transmission, you're probably selecting for better fitness, and therefore that could have other effects. So I love this.
I also thought that was a really key point to the reason this paper was very interesting.
Yes, it is. This will come back in the discussion, right?
Yes.
Yeah.
So this isn't really a lot about Legionella. It's about the phage of Legionella, and there aren't any reported phage that have been isolated and been studied. There were a couple of observed phage, but they hadn't been able to study them in the lab. And there was also a bioinformatic approach taken to look for Legionella phages, and they found that there were matches to CRISPR-Cas spacers that matched to microviruses, as well as to this integrated element called the Legionella mobile element 1, or LME1. This is a 30 kb region of the Legionella genome. And so those were the two prior reported clues that led us to think that there would be phages. And in this study, they were interested in activating and figuring out if this actually was a viable phage. And the reason for that is because phage are one of the natural environmental selective pressures that could potentially be driving Legionella to have traits that would lead to human disease. So in this paper, what they did was they used a particular Pneumophila strain called Mercia, and this had this LME1 element integrated into it, but it was not at a high copy number, and they couldn't really find any phage, what they thought might be phage propagation from it. So what they did was they took this interesting approach. They first tried the typical methods of activating a prophage that we would use. That would be exposure to UV, or exposure to another DNA damaging agent like mitomycin C. These turn on DNA repair mechanisms that are known to activate many prophages, but not all, and it did not activate anything in this case. So instead, they added a chloramphenicol resistance gene into this LME1 element, and then they passaged the Mercia Pneumophila strain until they had higher chloramphenicol tolerance, which would require either a higher copy number of the gene or a higher expression from that locus. And they saw that they did get higher copy numbers from that passaging. And then they started to investigate this, what they call the CMR passaged strain. And they took a couple of approaches to see whether or not they had expression of anything from that strain. So they first had an antibody against the major capsid protein of LME1, and they saw that if they just looked at their original strain, or the strain where they had added the chloramphenicol resistance cassette, or a strain where they managed to cure or get rid of the LME1 element, they didn't see anything on their blot. But when they had the passage strain, they did detect major capsid protein on a Western. They also looked for the presence of DNA from this phage-like element, and they used a restriction enzyme that could cut that region, and they just looked on the gel to see if they could see a banding pattern, and they only saw that when they had their passaged phage. So they took the supernatant from these passaged strains where you would expect that there is, and this is the case with most prophages, I'm assuming also proviruses, that there is a low level of activation occurring in any culture at most of the time. So they took the supernatant and they were able to, they tried making plaques. They did not have any plaques. So they tried to isolate virions and they ran these. They first concentrated them by PEG, very common method, and then they ran them over a cesium chloride gradient, and they had a band, a white band. I'm actually not clear why sometimes the bands of viruses are white and sometimes they're blue, because I thought that the blue came from the presence of DNA. But anyway, they had a white band, and
which I just have to say, as I was looking through this paper and I saw figure 1, and I was like, oh, they actually ran a cesium gradient. This is awesome.
There's a cesium gradient, and they did restriction enzyme.
Yes,
Yep.
yes.
Which I haven't seen in years in a DNA gel.
Yeah.
But my understanding, Jolene, is the blue is the same blue that comes from the sky, right?
Mm-hmm.
It's a kind of a Schlieren thing of particles, and that's why it's blue.
And but this one wasn't blue. I thought it's so it's not because of the DNA.
It looks like it lightly tinted with blue.
Yeah,
I don't know, but it's beautiful. I
agree with that.
You know, it's the tube with the sealed top. It's the whole thing. We talked about cesium chloride gradients when we did the Mendelssohn and Stahl paper not too long ago. It's very nice to see that.
Right. So they.
But Jolene, I wanted to know why. So they knew about this LME1 element for a while.
Yep.
Couldn't they sequence it and see signatures of phage proteins?
They did see that, but that didn't tell them that it would be active.
Okay.
Yeah. Because there are many incomplete elements that could have been transferred, and so it doesn't actually mean that it would lead to a phage particle. And a lot of these, especially if it's in a bug where you haven't seen a phage before, you get a little bit of hints that there's something, but you're missing a lot of the genes that you would expect to be present to have an active phage. So if you don't see a particle, then it could be an element that does not lead to an actual self-replicating virion.
Okay.
So they did mass spec on this purified material in their cesium chloride band, and they saw they saw 12 of the protein products that would be present in the LME1 region, including major capsid protein, tail tube proteins, the portal protein, tail fiber protein, and some internal virion proteins, and then ones where you wouldn't know whether or not, or what exactly they were doing.
I think this is great because in the day you couldn't do mass spec. You had to run protein gels. You had to cut out the proteins, try and figure out the sequence. You didn't get most of them because it wasn't enough. But boom, mass spec does it all.
Yeah.
It's amazing.
They also sequenced the DNA that was present in the band, and it was exclusively LME1 DNA. No chromosomal DNA, they say, did they find at all. So this suggested that they had intact virions. I think it's kind of funny that they don't just have a picture, but the very next thing that they show us is that they went to the microscope and they did cryo-EM to solve the structure. So they first show us these nice little short tail podophage virions, and then they show us the cryo, which is also very pretty. Nicely colored with the different parts of the capsid. So they've got their major capsid protein, and then they have a decoration protein around the outside. They show us a split where you can see the portal, an adapter, a tail fiber protein that wasn't as well resolved, probably because it's flexible, and then at the bottom, what they call the nozzle, which is slightly pointed. And they can also see the DNA inside of this virion structure, nicely spooled in layers, which would be a nice ordered way to be able to fit it all inside. It's a T=7 size, and it's also a T7-like, so similar to the E. coli phage T7. It's a kind of a way that we use to help us draw some comparisons with phages that are already known.
What I find interesting is that these surface proteins, they're not turrets like the phage, like the virus we just described, and they're not at the five-fold axis. They surround the five-fold axis. You can see in that panel A. towards the bottom, there is a single 5-fold axis that's
Yep.
very clear, and these, what do they call them? The proteins,
Decoration proteins.
The decoration proteins are surrounding it, maybe at the 2 or 3-fold axis.
Yeah,
That's cool.
They kind of look like pairs of doggy bones to me.
They do. Agreed.
So they say that there is a very narrow opening at the nozzle, which makes sense because you don't need to put a lot through there. And then, so now they've seen that they're making full, complete virions. They still could not make plaques, which we're finding is not that unusual for some environmental viruses that haven't been studied before. Traditional plaquing does not work. A little bit sad, but they had to come up with other ways to show that they were making virions that didn't involve, gradient and Western and so forth every single time. So the 2 assays that they used to show that they were making phage in any given environment was first an attachment assay. And in that attachment assay, they would mix phage with bacteria and then look for the signatures of phage just stuck to a bacterial pellet that they spun down. And then the second assay was what they call an infection assay to generate lysogens again. So they would use their Delta LME1 strain, which was basically the cured strain that they were able to isolate, and they would add this LME1 with the chloramphenicol cassette, and every time that it forms a lysogen, then those cells would become resistant to chloramphenicol, so they just could count colonies. And using these 2 assays, they first showed that if they heated the particles, then neither of those would give them a positive signal, which makes sense because when you heat the virion, it falls apart and then it can't infect. It can't attach because it needs its folded tail fiber to attach, and it can't put its DNA inside because it's missing pieces. It's fallen apart. So then they added this phage to the host, and they saw that it didn't really have a very big effect on the growth of the host in liquid media unless they added a lot. And even then, it wasn't a very big effect. It didn't inhibit the growth significantly. They did also, let's see, what did they do next? Okay, so then they started to look at the effect of LME1 outside. Am I skipping something here? Right. So they looked at the effect of LME1 on the host itself in terms of colonies that grew after exposure. So they exposed the Nemophila host to the phage, and then they picked colonies that were resistant, that still grew, and they sequenced them. And they didn't get very many. In this case, they sequenced only 6, and 2 of them had single nucleotide changes in a gene called LAG1, and then 4 of them had actually undergone a recombination event so that they removed completely that LAG1 locus from the genome. And they checked this delta LAG1 with their 2 different assays, and they saw that when LAG1 was not present, then they had the presence of the major capsid protein in their attachment assay, and they saw a high level of lysogen formation in their chloramphenicol selection assay. They saw that for a strain, a Philadelphia strain and a Toronto strain. So these are strains that are different from that original Mercia strain where they isolated the LME1 originally. And that suggested to them that the LME1 could infect and replicate in multiple different Legionella Nemophila, and 2, that this particular gene was going to be important in multiple different Nemophila hosts. So what is this gene? Okay, so LAG1 is the lipopolysaccharide O antigen O acetyltransferase gene, which modifies the LPS. And the LPS is the surface lipopolysaccharide of the bacteria. So it's gram-negative, and it has LPS on the surface. If you modify that, then this suggests that LME1 is dependent on the presence of that modification for its infection. Okay, so what they then wanted to look at is whether or not this was important in Legionella pneumophila's ability to survive inside of a macrophage, which is where it will replicate in a human when Pneumophila is causing disease. And in this experiment, let me find the experiment here. So they combined the Legionella pneumophila that did not have LME, and then they combined it with LME and showed that they got this LAG1 change. They put these into the macrophages, and they measured how much did they get out, how much replication was Legionella pneumophila able to undergo when it was inside of the macrophage. And this is what is shown here, where they see that when they have no phage added, then they have lots of growth inside of the macrophage. When they add a little bit of LME1, which would start to select for that deletion of LAG1 gene, then they have a reduction in the amount of Pneumophila that can grow in the macrophages. And when they add a lot of LME1 to the cells before they put them with the macrophages, then they see almost basically no intracellular replication for Pneumophila. And then they show that when they heat inactivate the LME1 phage, they recover all of the ability for Pneumophila to replicate inside of the macrophages. And they also see that lysogens, or the ones that have LME1 integrated, are perfectly able to replicate in the macrophage. So this is kind of the key experiment that leads to this idea that an environmental pressure, in this case the bacteriophage LME1, is selecting for the removal of this LAG1 gene. And when that's gone, then or do I have that backwards? I think I might have said that backwards.
Right. So the phage selects for removal or destruction of the LAG1 gene.
Right. And most of these strains that that will infect people can have an association of the presence or absence of LAG1 being associated with the ability to cause disease.
Right.
So presumably this selection happens in the soil, which is the natural place for Legionella to grow. And then if it somehow ends up in people, then you have this virulence phenotype, basically.
Right.
So the phage applies a selective pressure that selects for the bacterium that doesn't have LAG1.
Right.
And then when that bacterium gets into a human, that lack of LAG1 makes it better at infecting macrophages and makes it more virulent.
Which is a coincidental effect.
It's a coincidental effect. The thing I thought was really interesting here, though, is when I think about phage involved in human pathogenesis, I think of like scarlet fever or cholera toxin. They mentioned cholera toxin in the discussion, where the phage is bringing in some toxin that makes the disease worse. In this case, it's the reverse of that, that the phage is selecting for resistance to it, and the bacteria that don't have the phage, that have acquired the ability to resist the phage, are actually the ones that have become deadlier or nastier, because coincidentally, they've gained this ability to grow better in macrophages.
Yeah.
Exactly. Yeah. So there are some phages that are usually temperate that can integrate, and they encode a toxin, and that toxin can lead to disease. The cholera toxin is the really commonly cited example. There are many other temperate phages with potentially toxins, and we don't like to talk about those very much, but they do exist, and that's why it's really important to know what's in a phage before you start doing things with it. And here, it's the phage doesn't even have to be there anymore, because it already led towards more virulence. I think the field has really started to think about this a lot in terms of choosing phage that can shift a population away from virulence, because that does occur. But this is an interesting example, and I think a very important example of when the phage can shift fitness traits towards what would be virulence for us.
Yeah, they mentioned in the discussion that when you're doing phage therapy for bacterial infections, you need to keep that in mind, right?
Yeah. I think we hope to do it the other way, but if we want to do it the other way, we have to do it very carefully.
I guess you need to do some biology in the lab to see if this can happen. Yeah.
Right. So the last experiment is where they took a panel of Nemophilus strains, some that they call Pontiac strains and some that are non-Pontiac strains, and they have a series of monoclonal antibodies that help them to know what kind of O antigen acetylation they have. So this is an indicator of their LAG1 status or genotype. And then they added the LME1 phage to see which ones would be able to attach. And they saw that the Pontiac strains, which would be the ones that cause a less severe disease in humans that doesn't always progress to pneumonia, those ones, they did not have attachment. They didn't have major capsid protein of LME1, and they didn't form very many, if any, lysogens at all. But the non-Pontiac strains, they saw a complete association where LME1 major capsid protein was present. And here they didn't see a perfect correlation with lysogen formation. There was 3 that did have high levels of lysogens and 2 that did not. So that indicates that there's probably more going on in those particular cells where lysogens were not formed, suggesting there's some sort of restriction or defense that those strains are encoding against LME1. Okay. And so that's the last experiment, and we kind of already talked about the model, but just to re-summarize, so in Legionella pneumophila lives in the environment, in aqueous and soil environments. It infects amoeba and other protists. It is going to be experiencing those environmental selective pressures as well as potential exposure to phages like LME1. This has caused Legionella pneumophila to acquire a large repertoire of effectors that it uses to successfully infect these different hosts that it grows inside of. And those allow it to then also grow inside of macrophages, where it will invade, it will survive inside of a vacuole and use those effectors to cause disease and replicate. Similarly, the phage resistance will select for a change in the status of the modification of the O antigen on the LPS on the surface of the bacterium. And when it does that, what they are, I think, proposing for the most part, they have shown this with some of these strains, that the change in the O antigen status allows for resistance to complement-mediated killing. So that wasn't shown in this paper, but that is shown elsewhere. And that is what they're calling an accidental virulence trait. Just like the intracellular replication in the macrophage, that's not its native host, but it can do that because it does that in the environment, in the amoeba and other protists. And just like in the environment, it is selected away from having this LAG1 locus active, it also then will be able to resist complement in the human host. Yeah. And I skipped a little bit of the experiments where they talk about deletion of CRISPR, but they also show that there's a little bit of a synergistic effect. When you remove both LAG1 and CRISPR, then the cells have an even greater effect in the face of the phage. So that's the major story, is that environmental selection pressures will change what the host cell looks like, and that can have consequences that you might not have anticipated. The phage here is selecting for Legionella pneumophila that are more likely to cause severe disease in humans.
So this experiment where they looked at these 10 isolates, I think,
Mm-hmm.
they found 5 that encode lag and 5 that don't. they don't mention which ones were virulent or not, right? Because it could be that the ones that encode that lack lag, right?
Mm-hmm.
Those would be less virulent. That would be how it goes, right?
No, those would be more virulent.
So they lack lag, therefore they can be they're not infected with the phage, right?
Right.
But the selection for resistance to phage is accompanied by an increase in virulence.
Yeah.
Right.
Mm-hmm. Right.
So but anyway, the point is they don't say which are virulent in them because you would predict that
they're all isolates from humans that were infected. So they're all causing disease, but I understood that the Pontiac strains are less virulent. They cause a less severe disease, and the Pontiac strains cause a more severe disease.
So those...
sorry, did I say that backwards?
Those should be susceptible to the phage.
The Pontiac strain's less severe.
Less severe.
And the
non-Pontiac
strain's more severe.
So the less severe ones should be susceptible to
phage.
Which would mean they lack LAG1. I'm sorry, they lack LAG1. They're susceptible to the phage because the ones that encode LAG1 are resistant to phage.
No, I think that's
the other
backward.
way around.
No, it says right here, the 5 strains that encode lag were resistant to LME1 attachment.
That's true.
So the LME1 modifies the outer surface so that the phage can't attach.
Oh, yes.
Sorry, LAG1, yeah. Anyway, so that you would, if you got more and more isolates from mild versus severe cases, that would be interesting to look at the correlation with lag, right? Lag plus or minus. That's all I'm saying.
Yeah.
And maybe other things too could do it, not just lag, right?
Yeah.
That's right. It is not just lag.
Yeah. It's very nice. I like this. This whole, as I said, that whole coincidental virulence is just great. I love it. I always liked the word Pontiac for some reason.
Has a nice sound to it.
I just looked it up. It was the name of a First Nations chief in the area. And so the car company took it to name its car after this guy. And the city, I guess the city was named first, and then the car company took it after the city. Yeah. But the city was named after the chief. It just has a nice feel to it, right? Some words just have that nice feel, Pontiac. But, I can't help that stinking car come into my head.
Right.
Never thought it was a very good car. Thank you, Jolene. A tag team of, we can't call them viruses. They're all viruses.
They're all viruses. They're tailed viruses.
Archaeal and bacterial viruses with tails, TWiVs. Okay, let's, it's 4:24. We could do a little bit of email here.
Okay.
Let's see. Okay. Alan, can you take the first one?
Sure. Anonymous writes, Dear Amazing TWiV team, several CDC colleagues of mine, scientists who care about truth and science, fed up with the impunity with which this administration is acting, used Bluetooth speakers to ridicule and mock HHS Deputy Secretary and Acting CDC Director Jim O'Neill during a meeting of the conservative anti-science American Association of Physicians and Surgeons, and provides links to a video of the event. They did this in hopes that we can break the sense of normal decorum when the administration is acting so abnormally, and directly communicate to the face of HHS, CDC, and NIH leadership that they will be remembered for making a mockery of America's long history of scientific progress and medicine. In truth and science, a humble servant. Thank you. This is definitely funny to watch.
It's true. It's a mockery. They're making a mockery.
And so this whoever arranged this prank made a mockery of them, playing circus music over the Bluetooth speakers while the guy was giving his talk.
I see. Wow.
Jolene.
Yes, David writes, the insecure son of a famous father, RFK Jr., seeks fame at any cost to anyone and anything. This is the inevitability of Greek tragedy playing out once again, as if the narcissistic son in the White House were not enough. In any other setting, they would be mere buffoons and fools, comic actors on a Shakespearean stage, idiots telling tales, full of sound and fury, signifying nothing. But they are real. David Davin.
What Shakespeare is that from?
Oh, it is a tale told by an idiot, full of sound and fury, signifying nothing. Is it a Hamlet?
Let's see. Macbeth.
Macbeth. Yes.
Thank you, Google. I think this is a perfect application of that line, isn't it? Buffoons and fools, comic actors. Thank you, David. That's
Macbeth
great.
is not a comedy, though.
No, it is not.
Everybody does.
That's the thing.
It's, yes, that.
This is real life.
This is not a comedy either.
John writes, and with respect to COVID surge of late summer, I suspect that it is Willis Carrier's fault. Who's he? Invented the air conditioner in 1902. When the heat gets intense, people are more often inside. Just look at what regions lead in summer versus winter on the wastewater plots. In summer, it's the South and West. In winter, it's the Northeast and Midwest. And they trade places at those two times. This must have been noted before, but I don't remember it. I would say that's also for Legionella, right? You use your air conditioners, you get water in it, boom. And RE wastewater, Elke Volants was great. And Alan, Swedish pronunciation guide. The
Umio.
Umio. I don't know what all these ampersands.
Yeah,
things.
The characters didn't translate well. Linköping and Lund.
Okay, so Linköping is as Linkopping, which would be Linköping.
Linköping.
And Lund is there, and Umio is just. Is it UME or UMEO?
It's just UME.
Umio, yeah. Regards from a Greater Braddock that was uncommonly pleasant today with a high of 19C combined with rational humidity.
Rational humidity, wow.
That's an interesting one.
I don't think I want my humidity gaining sentience.
Alan, can you take the next one?
Sure. Anthony writes, as with theory, the problem is that people use the words feel, instinct, gut feeling, and gut instinct to mean very different things. One, any stray thought that comes to mind. This can include passing delusions like Feynman and the ringing phone that for a moment he thought meant that his grandmother had died. Imagination is the fountain that both science and art draw from. The scientist tests ideas by experiment, observation, and calculation. The artist puts the mental raw material on a métier forge and hammers away to achieve perfection. Both practitioners know that inspiration is the beginning, not the end. We speak of the instinct of a police professional or Gladwell's art historian in Blink, gives a link. Here, instinct is anything but. It's automatic processing born of training and long experience. Third, actual animal instincts, by definition archaic, persisting in humans. These are few and can be worse than useless. Boxers are constantly cautioned not to act on instinct. When threatened by a striking snake, taking a step back is a good idea. If a fighter is punched, stepping backwards means getting punched again. For a non-human primate losing balance in a tree, reaching for a branch to grab makes sense. For someone slipping on a sidewalk, an outstretched arm is an invitation for snapped fingers, a broken wrist, a fracture in the arm or shoulder, and cartilage damage. For what it's worth. Thank you, Anthony. And then he gives several quotes from Trump talking about how he feels that vaccines are causing harm.
I feel very certain.
Yes.
I feel. My God.
No feelings allowed in data interpretation.
No. Jolene.
Yes, Terry writes, hello to Vincent, Brianne, Rich, Alan, Angela, Kathy, Jolene, and the spirit of the late, great Dixon. First, I want to thank all of you for continuing to give the world high-quality science communication. I discovered your podcast with my now ex, and while that relationship sadly did not work out, I remain an avid listener to TWiV.
Oh, my gosh.
Well,
are we causative or an association?
I think we are a healthy long-term relationship.
Agreed. Second, I just listened to episode 1259, and I was heartened to hear what you all had to say about AI. Though I'm not a scientist, I have an active interest in philosophy. This is AI, right? Or is it? Okay. in philosophy, anthropology, psychology, and history. I have a few observations and comments to make, though upfront I agree with all that you said in the episode. AI is a nomenclative error which derives from science fiction writers in the 50s. The technology as it currently exists would be better called applied statistics, because an elaborate autocorrect is all that it is. The problems that exist in the science and philosophy of consciousness are so intractable that we lack consensus about almost everything about it within those fields, which obviously makes producing and reproducing it artificially completely impossible. Because it is an elaborate autocorrect, it frequently produces hallucinations which have no relation to reality because it is all about the statistical likelihood that a given word will follow another. The problems with using, quote, AI in science go beyond merely producing statistically probable sentences. It's been shown that even experts will start to distrust their own judgment if it contradicts what the computer says, something which is dangerous for obvious reasons. You also point out that there are too many unknowns, and this is absolutely true. To add to this, nature specializes in what Nassim Nicholas Taleb calls black swan events. Donald Rumsfeld would call them unknown unknowns. Nature loves to do things so new that we not only don't know what happened, but we don't even know what questions to ask to find out. I agree that there may be limited and genuine use cases where, quote, AI can potentially be helpful. But as you say, it can't replace laboratory work that uses real animals. It can't take the place of field research. It can't really do anything that has real-world bearing and import. And because those cases are precisely the ones where a human dedication to truth and the ability to understand, quote, truth as an abstract concept, something that, quote, AI cannot do because it is not consciousness, it is not conscious, and a human attention to detail and a human imagination are at their most necessary, and the propensity of, quote, AI to conflate the statistically probable with the true is at its most dangerous in those cases. Once again, thank you for all the excellent work you do. Yours, Terry.
Oh, that's very interesting.
Yeah, well put.
So it's really a statistical application of.
Yeah.
Oh my gosh!
I mean, most of the time when we're talking about quote unquote AI, we're really talking about application of machine learning models, which are really a lot of complicated and, sometimes very accurate math.
But they're not consciousness.
Yeah.
Well, that's why you use them to do protein structure predictions, right?
Yeah.
Because you feed them what we know, this amino acid sequence folds like this, and they make a prediction. And it could be right or not, right? You
and
can
it
test
is a prediction, yes.
It's a prediction, exactly.
And it should always be labeled a model, a prediction.
Yeah. In fact, in the archaeal virus paper, they say they used AlphaFold to make predictions, and then they went to their data, and it helped them to model what they saw. So that's a great use of it.
Sure.
Right. But I think we were talking about it in terms of predicting what virus would be the next pandemic, right?
Or replacing clinical trials with AI or
or animal models with AI. I mean, I'm not being a Luddite, but I just don't see how it works. Given
you can't replace the models with this. You can use it to. You might be able to cut down on the number of animals you need to use by doing experiments that are more targeted.
Yeah,
it can give you hints. It can give you predictions. You can check those predictions, but it's not going to give you answers.
It's an elaborate autocorrect. Isn't that great? What were you going to say, Jolene? I'm sorry.
It's like Terry. Terry says, the more new things we do, the less useful it is. So when you're doing things that have already been done, that you just need to repeat with a slight variation, then it's really great. But if you're asking really, truly novel questions, still need the people.
It's an interesting debate, right? Or discussion.
Sure.
I like because it makes you understand things better. All right, let's do some picks of the week. Alan, what do you have for us?
I have a Science News article by Eric Stokstad telling the story of the last female great auk. Now, the great auk was known as the Arctic penguin, even though they're not closely related at all. But this was a large flightless bird, aquatic bird that swam around in northern waters and nested on land. And being large and flightless and having, this interesting plumage, it was an easy target for people who ships would stop off at islands that were nesting sites for great auks, and the crew would go ashore and just kill a bunch of great auks and stock up on poultry. People thought they were plentiful, and they were. These were a dominant species across the North Atlantic. And then I think there was a craze for the feathers at one point for fashion. So then professional hunters were showing up and just collecting them by the tens of thousands. And they went extinct. It's one of these horribly sad stories of human-driven extinction. These big, beautiful birds that are now gone forever. The final pair that was known to have existed got killed by some people who were hunting them. And ended up, at that point, I think they were mostly being stuffed and sent to museums because they were so rare. So they were driving the extinction because they were less common. It was known where this last pair, this nesting pair, the male of the two had been found in a museum, and there was some confusion about where the female specimen had ended up. But this is the story of how that was all figured out, and it is now, in fact, in Cincinnati. And was in a storage room, because museums have big storage rooms for things that are not on display, and they're now planning to put it back on display.
So the dodo bird had a similar ending,
yes.
Right?
Dodo had a similar ending. The moa, I think
moa,
it was
right.
in New Zealand. That was this huge ostrich-like, not related to ostriches very closely, but enormous bird hunted to death.
So they call the last two of a species the endlings.
The endlings, yes.
And they say the last passenger pigeon died at the Cincinnati Zoo.
Yep.
And the last Carolina parakeet died in Cincinnati.
Don't go to Cincinnati if you're a scarce species.
So there's a book here that they cite, which sounds very interesting. It is by Dolly Jorgensen, a historian at the University of Stavanger. She wrote Ghosts Behind Glass, Encountering Extinction in Museums.
Yeah.
That could be interesting.
It could be.
Wow. And depressing.
It's very sad that these guys go on this island and they strangle two great auks. They crush the egg. What? Come on.
Well, that was their job, was to go and...
Yeah, I know, but
that doesn't
in
make
retrospect,
it okay.
it seems horrible, right?
Yeah.
Jolene, what do you have for us?
I picked something called Ribosome Studio, which is an account that periodically pops up on my Instagram feed. It's just so beautiful. These are scientific animations of molecular sceneries. And sometimes they will model an enzyme. Sometimes they'll model a structure of a protein and its movements as it is doing whatever it does. Sometimes they model cells. I just think they're so pretty. They have a lot of them that are, eukaryotic cells. I love the ones with membranes. They'll show the lipids and they'll show them moving around and the proteins in there. It really illustrates how dynamic things are inside the cell. I think my favorite one is the one of the ATP synthase that shows the rotation that's occurring as protons are moving across the membrane through the channel and how ADP is converted into ATP. I love these kinds of things that show the movement of proteins as they're folded in their structure in the native lipid environment. So
that's cool.
This is Ribosome Studio on Instagram.
That's very neat. Beautiful. I just subscribed. I just followed, Nice. Now, my pick, the original article in the Times is paywalled, so screw that. I'm going to go right to the science article, which is also paywalled, and I can't read the whole thing. But I'm going to give you the gist of it because it's really interesting. This is a research article, Greater Noctule Bats Prey on and Consume Passerines in Flight. So these bats catch birds in flight using echolocation and eat them on the fly. And not only that, so the technology here used to do this, they use high-resolution biologging tags to show that these bats go to high altitudes, engage in long echo-guided chases, and consume the birds in flight. So they use a private sensory channel and echolocation to do this. And they also go to high altitudes where nobody else goes.
Except the birds.
Except the birds. So they say there's a rich food resource way up there, but most predators can't get to it. Now these bats have figured it out. And they've been doing it for a while. It's nothing new, but we just got the science to be able to.
We finally caught them in the act.
And we got the funding too. But I think that's really interesting, right?
And passerines are the perching birds. They're half of all birds. They're the birds at your bird feeder. And lots of them migrate, and the ones that do tend to fly a lot. Often they'll fly at night. So this, they're cruising. The first robin of spring is on its way, cruising at a relatively high altitude, and
intercepted.
Bat intercepts it.
It's very interesting. Anyway, the authors are from places in Spain, institutions in Spain. I thought that was really interesting.
Yeah.
And I couldn't read it, but I actually... Yeah,
you don't have access to science?
I don't have access to science, no.
Oh. Columbia should have
So I shouldn't say I don't have access to science. I do, but you guys don't. The listeners don't.
Okay,
so
okay, yes.
I hate to pick things that
I have press access to it. And
yeah. I mean, I can find an article by going to the Columbia libraries, and
right.
It's a bit of a
hassle,
a pain in the neck. But I also have to get a dual thing on my phone, to log to make sure it's me. And then it's just hassle.
It's the same for me when I'm going into the press.
But if I'm doing a paper here or on any podcast, I will go through that and download it, obviously, because I have to read it. But for a pick, I don't want people to have to read it, so I will explain it. So that's it. It's a really interesting.
There were a bunch of news articles about this. So if you get the Times, you could read it there. If you read some other site, you can probably find this news online.
So I saw it.
It is a cool story.
Where did I see it initially? So I used a feed reader to get stories.
Yeah, I saw it a few different places.
And I saw it in the Times, and then I said, screw that, and then went to the science. But there you go. It's a cool story. All right, that's TWiV 1261. You can find the show notes at microbe.tv/TWiV. If you want to send us a question, a comment, or a pick of the week. We don't have any more recent picks. I have ancient picks. Most of them are not interesting anymore. But send them in, twiv@microbe.tv. If you enjoy these programs, we'd love to have your support. And also, if you were in the New York area, in December, we're going to be doing live here at the incubator Immune number 100. We have room for about 20 people here. So we'll be announcing it this weekend. You're going to have to buy a ticket to get in, but it should be fun. We're going to have the whole Immune crew and a couple of guests, and we're going to give you some stuff to take home.
How exciting.
Cool.
It will be announced on the social channels?
Yeah. It will be announced on the normal places. So, yeah, microbe.tv/contribute. That would be how you can help us out. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Jolene Ramsey, Texas A&M University, ramseylab.vercel.app. Thank you, Jolene.
Yeah, this was a lot of fun talking about tailed viruses, TV on Microbe TV.
Yes.
Ooh, look at that. Yeah. Just 2 tailed viruses today. This is probably the first time it's ever happened in the history of TWiV. 1,261 episodes. Yeah, I know you don't care, but we care. We think it's
sure. Yeah.
The things we think are cool, you may not think are cool, but one day you will when your tastes become more sophisticated, or I should say nuanced. I'm Vincent Racaniello. You can find me at microbe.tv. And Microbe TV is on Blue Sky. microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV. Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.